Chapter 14, Section 3, Studying the Human Genome. Our key questions for this section: what techniques are used to study human DNA? What were the goals of the Human Genome Project, and what have we learned so far? Roman numeral 1, Manipulating DNA. Even the smallest human chromosome contains nearly 50 million base pairs. When we're talking about the base pairs, we're referring to adenine, thymine, guanine, and cytosine. Number 2, what techniques are used to study human DNA? By using tools that cut, separate, and then replicate DNA base by base, scientists can now read the base sequences in DNA from any cell. We take a look at the video here. Nucleic acids are chemically different from other macromolecules such as proteins and carbohydrates. The difference makes DNA relatively easy to extract from cells and tissues. However, DNA molecules from most organisms are much too large to be analyzed, so they must first be cut into smaller pieces. Many bacteria produce enzymes that do exactly that. Cutting DNA. Known as restriction enzymes, these highly specific substances cut even the largest DNA molecule into precise pieces, called restriction fragments, that are several hundred bases in length. Of the hundreds of known restriction enzymes, each cuts DNA at a specific sequence of nucleotides. EcoRI cuts anywhere the sequence GAATTC occurs. In this example, the sequence GAATTC occurs 2 times. EcoRI cuts each strand of DNA between the G and A bases, leaving single-stranded overhangs with the sequence AATT. The overhangs are called sticky ends because they can bond or stick to a DNA fragment with a complementary base sequence. Separating DNA. Once DNA has been cut by restriction enzymes, scientists can use a technique known as gel electrophoresis to separate and analyze the differently sized fragments. A mixture of DNA fragments is placed at one end of a porous gel. When an electric voltage is applied to the gel, DNA molecules, which are negatively charged, move toward the positive end of the gel. The smaller the DNA fragment, the faster and farther it moves. When the electric voltage is turned off, the DNA fragment stops moving and can be viewed in the gel using ultraviolet light or staining. Reading DNA. After the DNA fragments have been separated, researchers use a clever chemical or trick to read or sequence them. The single-stranded DNA fragments are placed in a test tube containing DNA polymerase, the enzyme that copies DNA, along with the 4 nucleotide bases A, T, G, and C. As the enzyme goes to work, it uses the unknown strand as a template to make one new DNA strand after another. The tricky part is that researchers also add a small number of bases that have a chemical dye attached. Each time a dye labeled base is added to a new DNA strand, the synthesis of that strand stops. When DNA synthesis is completed, the result is a series of color-coded DNA fragments of different lengths. Researchers can then separate these fragments, often by gel electrophoresis. The order of colored bands on the gel tells the exact sequence of bases in the DNA. The entire process can be automated and controlled by computers, so that DNA sequencing machines can read thousands of bases in a matter of seconds. The base sequence shown here as read from the order of the bands on the gel from bottom to top is T, G, C, A, C. Roman numeral 2, the Human Genome Project. Number 1, The Human Genome Project was a 13-year international effort with the main goals of sequencing all 3 billion base pairs of human DNA and identifying all human genes. There were other important goals as well. 1. Sequencing the genomes of model organisms to interpret human DNA. 2. Developing technology to support the research. 3. Exploring gene function. 4. Studying human variation. And 5. Training future scientists to be able to continue their project. Markers. Markers are used to break up the entire genome into manageable pieces, kind of like a mile marker on highways. Comparing sequences. If you were to compare the genomes of 2 unrelated individuals, you would find that most, but not all of their DNA matches base for base with each other. On average, one base in 1200 will not match between 2 individuals. The International HapMap Project is to locate and identify as many haploid types in the human population as possible. It will give scientists a rapid way to identify haploid types associated with various diseases and conditions. Number 6, the Human Genome Project was completed in 2003. This was the 50-year reunion after Watson and Crick published their DNA structure. Number 7, bioinformatics. This is the application of mathematics and computer science to store, retrieve, and analyze biological data. We take a look at the picture here. It is a new field that combines molecular biology with information science. It is critical to studying and understanding the human genome. Number 8, genomics. This is the study of whole genomes, including genes and their functions. Number 9, the Human Genome Project pinpointed genes and associated particular sequences in those genes with numerous diseases and disorders. It also identified about 3 million locations where single-base DNA differences occur in humans. Number 10, this can help us find DNA sequences associated with diabetes, cancer, and other health problems. Number 11, ethics. In 2008, a new law was passed called the Genetic Information Non-Discrimination Act. This was passed by President George Bush. Lots of questions arise about ethics and genetics, such as who should have legal rights to your genetic information? If you had your genome read, who should or should not get that information? Think about, would you want your employer to have that information? Would you want an insurance company to have that information? Why or why not? And number 12, what's next? They are working to better understand the human genes. In our book, it talks about the 1,000 Genome Project, which was launched in 2008 to study the genomes of 1,000 people in an effort to produce a detailed catalog of human variation. This project is continued and is being worked on, but is also about 12 years old. They're doing many more recent projects, and so I'm leaving you with this last question of, do some of your own research. What are they doing with the human genome? What kind of projects and studies are they working on currently? I'm going to add a question into Google Classroom for you to answer and just add a little bit of information of what you found, where they're taking these studies, and what some of the new things that they're finding with this information. And that concludes section 3 of chapter 14.